All right, so I'm going to try to keep these videos a little shorter, but the next thing that I wanted to talk about is the actual makeup of the genetic code in an organism. And what we've talked about so far are these little subdivisions of a chromosome called genes. A gene is a segment of DNA that controls a specific trait, and if you change the order of the bases, the letters of the genetic code inside of the DNA, then what you get is a different version of a gene. So maybe a different eye color, maybe a different hairline, or Papa Smurf. Now in this case, it's time for me to let you in on a little secret. Not all of your DNA is genes. If you get to the end of one gene, you might get to another gene, or you might get to highly repetitive sequences. These vast stretches of your DNA that are just the same few bases or letters that repeat over and over and over and over and over and over and over again, and they don't give you like a trait of any kind. So that's a little weird. The analogy here would be like if you were reading a book and at the end of a chapter, like a gene, you just get to a page or a few pages that have like a word on them, and that word is just going to happen again and again and again and again. Or maybe a whole sentence that just goes over and over and over and over and over again. It doesn't contribute to the story, it doesn't have anything to do with what's in the book, but you have to go through all of that before you get to the next chapter, or gene. So we're going to look at the difference between genes and these highly repetitive sequences. And so the first thing that I want you to know is that the single copy genes are what we would refer to as coding sequences. These are going to be the pieces of DNA that give you the instructions for a protein or a polypeptide. And in that case, these are the parts of your DNA that are going to give you a trait. The highly repetitive sequences do not code for a protein, they do not go through that central dogma, and they're not going to end up giving you a trait. Now, ultimately, this is only about 1.5% of your entire genome. That's not very much. A relatively small portion of your DNA actually gives you traits. And again, as we've talked about, inside of this DNA, you're going to have codons where every 3 bases tell you what one amino acid you're going to be putting on. Now, the trick here is that after you go through the central dogma, you take your DNA, you use it to make some RNA. That RNA might actually get changed up a little bit. And in this case, it's important to look at the difference between a piece of unchanged RNA and the finalized RNA. What's going to happen here is the DNA is actually going to contain regions that you want and regions that you need to cut out and not include in the instructions. So what we have here are introns and exons. Now, the names here are a little counterintuitive to me. When I think of introns, I think of something you're including or something that you're going to use. But that is the opposite here. Exons are the parts of the DNA that you want. Introns are the parts of the DNA that you're going to end up not using. So the first thing you do is you take your introns and your exons and you turn all of that into RNA. Then you chop the RNA up, you get rid of the introns, you splice together the exons, and you get a piece of RNA that has only the instructions in it you want. None of those filthy introns messing up your genetic code and having letters that you don't actually want in the instructions for your protein. So this actually falls into a category called post-transcriptional modification. You've done transcription, you've taken your DNA, you've made it into RNA, and then you chop the parts of that RNA out that you don't need. The rest of the RNA that you have is going to be called just the exons. And the reason you do the chopping when it's RNA and not DNA is that you don't want to chop up DNA. You don't want to change the DNA. So no chopping or changes to DNA. That code has to stay intact and unchanged as much as possible. So there's a picture in your learning targets that looks something like this. Hopefully you can tell me what is going on here to make your final segment, and you can tell me what the little parts here would be called, and ultimately you could tell me whether or not what we're doing here is DNA or RNA. So the next part that we're going to be looking at here are the other things in your DNA, the non-genes, and again we call those highly repetitive sequences. These are going to be parts of your DNA that some people think of as junk, Sometimes it's also called satellite DNA, but they're just sets of letters somewhere between 5 and 300 bases. And that 5 to 300 letter word is just going to happen over and over and over and over again in between your genes. Now, this actually makes up a significant portion of DNA, somewhere between 5 and 60%, depending on the organism. So in humans, it's roughly 60% of our DNA. Now, ultimately, these are non-coding sequences. These do not give us a protein at the end of the day. But they're not entirely useless. They do a couple of things for us, even if they don't give us traits. So, they can actually produce other kinds of RNA, not the mRNA I was telling you about before, where you cut out all of those introns, but these other things that we would call tRNA and rRNA. So maybe you make some RNA, but it's not the kind of RNA that you're going to use to build a protein with a ribosome. They also regulate gene expression. So using the DNA, which genes you use and which genes you turn off, silence, or don't use, that is actually controlled by these repetitive sequences. It's also really, really important for genetic fingerprinting, or what you might know as forensic identification. It turns out that if you get a mutation in your genes, that could really affect you. It could affect your proteins, your traits, and even your survival. Maybe you don't survive as well. But if a mutation happens inside of your repetitive sequences, no big deal. It's a mutation that really doesn't show up for you or have any bad effects. And that means that they accumulate over time. Over many, many generations, you and your family lineage have mutations that have been silently sitting inside of your repetitive sequences, and they are mutations that only you and your family lineage have. So this is the part of the DNA that people look at when they are trying to identify somebody. Maybe a person has been found, they need to identify it, or maybe we need to establish some relationships, determine if somebody is the father or the mother, long lost kids, see if two people are actually brother and sister or twins. So in this case, it is that part of the DNA that you would actually be doing sort of forensic genetic testing on. And again, it's because this part can have mutations that have shown up for generations and generations and generations without any ill effects. So this table does a little bit of a side-by-side summary. Again, your genes, or what we'll call single copy genes, only about 1.5% of your DNA compared to 5 to 60% in your highly repetitive sequences. These have codons. Every 3 letters over here codes for an amino acid. Over here, this is thought of as junk DNA, satellite DNA. They originally called it junk, but it actually turns out it's not junk. It does have some jobs. Single copy genes are going to have introns that you cut out and exons that you splice together. Over here, what it's made of is just these repeating words of DNA, 5 to 300 base pairs for each word. Over here, you're going to use this to make mRNA, and that's ultimately what you're going to use to make a protein. Over here, these do not make RNA that goes to ribosomes. They might make other kinds of RNA. They also regulate your gene expression, which genes you use and don't use, and ultimately you can use them in forensics for what's called DNA fingerprinting. And that is the end of subtask 3.